Okay, so today we're going to continue our topic on genetics. And previously we've talked about Mendelian genetics, and we talked about monohybrid crosses when we cross two traits. Okay, we get a genotype and we get phenotypes and of our offspring, possible offspring. And then we also talked about dihybrid crosses previously, which is also part of the Mendelian genetics. But remember, that was two traits, and we had 16 squares in our Punnett square box, and it was quite complicated, and you got to do the FOIL thing. Well, now we're taking it an even step above that. No, we're not going to have like 64 boxes or anything like that. Crazy. But we're going to talk about the literal gray area of genetics, and we call this non-Mendelian genetics. And it's super interesting because a lot of our genes are associated with non-Mendelian genetics and what we call polygenic, which is multiple genes, or polyallelic, which is traits that are found on multiple different alleles and genes. So let's talk about it. So we're going to talk about today the exceptions to Gregor Mendel's laws. Okay, so we're literally talking about the gray area. So, and you'll see what I mean by when I say that. So for some of these, some alleles are neither dominant nor recessive. And this kind of plays into our hair color, our eye color. The majority of our genes that have more than two alleles, which is kind of like brain explode emoji, but like, where did we get all these alleles from? Because they're on multiple genes, right? And so we have multiple alleles that code for hair color. So we have different variations or a spectrum of color for hair color. And we see that in humans, and we see it a lot in other animals, and we're going to talk about that today. And some traits are controlled by more than one gene. We call that polygenic, okay? And that means we're going to have multiple genes that code for the same trait. Hair color is one of them, and so is skin color and eye color. And we're going to talk all about that today. So let's talk about non-Mendelian genetics. So non-Mendelian genetics. So over here on the left, okay, we have Mendelian genetics, what we call complete dominance, okay? So remember, if you were homozygous dominant or heterozygous, guess what? You were blue. In this case, you were blue because blue is dominant to yellow. Well, if you are only yellow if you have two recessive alleles for yellow, okay? So this is called complete dominance. Now, and this is more what we see in Mendelian genetics, right? Heterozygotes and homozygous dominant, guess what? They share the same color, right? They have the same phenotype, okay? Whereas if you're recessive, you have the only time you express that phenotype, that recessive phenotype, is if you have two recessive alleles, okay? Then we have codominance here in the middle and incomplete dominance. Let's talk about incomplete dominance first. And over to the side on your note sheet, if you have it, I want you to put a little parentheses and write blend of color, because incomplete dominance is where we have one blue parent and one yellow parent, and all of their offspring are going to be green. So we have a blending of color. And how I like to remember this is neither allele is dominant to another. And guess what? Your offspring will most often not look anything like your parents. So I want you to ask yourself this question. Are there people that you know that look nothing like their parents? The answer is probably yes, because we have a lot of incomplete dominance genes in our, like, as humans. Like, a lot of them are incompletely dominant. Some of them are incompletely dominant and codominant, which we're going to talk about next. So codominance is when you have a blue flower and a yellow flower, and you cross a blue flower and a yellow flower, and they individually express, their offspring will individually express blue and yellow petals, individually express both phenotypes, okay? So whereas incomplete dominance is blending, codominance is we're going to express both of those traits or alleles. We're going to express those alleles individually. So those phenotypes will be expressed individually. And I like to think about codominants like co-pilots, okay? Both are pilots, both have control of the plane. Neither, well, I guess I should say this. Both are dominant, but not one is not dominant over the other, but both are dominant. Whereas incomplete dominance, neither are dominant. So let's write some stuff down. Let's talk about it a little bit more in depth. Okay, but first we're going to talk about how the environment kind of shapes our genes. And we kind of talked about this during evolution too, but not all genes happen with our gametes, right? Like it's not all passed down. We have some things that are called epigenetic, where our environment has shaped us. So I want you to think about identical twins. Their DNA is not 100% exactly the same. It's really close. It's about 97%, but there's 3% that is dependent on that environment. And the gene that must be present along with the proper environmental factors. Okay? So, that being said, remember your phenotype is your physical characteristic, and it's the result of the interaction between genes and the environment. Okay? Because you're not going to be able to survive as a dark colored rabbit, right? If you live mostly in an environment where there's mostly snow. Okay? So we kind of get really we're going to touch back on evolution after we talk about this today. But your environment can shape your population's genetics, and we call that population genetics. It's a whole thing. We usually do teach it in an advanced level class. However, this year we are not. But it is very, very interesting to see allelic frequencies change, and we can literally see the change happen due to populations evolving over time. And it's very, very interesting. So let's talk a little bit more about incomplete dominance and what we're talking about today, because it's mostly about the gray area of genetics. And incomplete dominance is our literal gray area, right? It's our blending of color. Okay. So incomplete dominance is when traits are not controlled by a recessive or dominant allele. Okay. They are neither are recessive or dominant. It's going to be a blending of color. Okay. And in some cases, the phenotype, the offspring will have the phenotype in between the phenotypes of the parent. Remember, like a blending or mix of color for that offspring. Okay. So a lot of times we see the phenotypes of the offspring do not even match at all what the phenotypes are of the parents. Now let's talk about flowers. Now we like to talk about flowers a lot because we see incomplete dominance and codominance a lot in flowers. And so it just makes it super easy. So we have a homozygous red and a homozygous white. If we cross a homozygous red, and we are going to say homozygous red because remember, we don't have an allele that is dominant or recessive. Okay? Both of these alleles are both of these genotypes, so homozygous, we wouldn't say homozygous dominant for red because red is not dominant over white, okay? Neither allele is dominant over the other. So we call it homozygous red. And if a homozygous red flower is crossed with a homozygous white color, all of the offspring will be pink. That's how you know it's incompletely dominant, okay? But like I said, incomplete dominance, alleles are neither dominant nor recessive. So when we say the genotype, we say homozygous red, which I know sounds crazy because you're like, oh my God, brain explode emoji. We were talking about like, that's like a genotype and a phenotype mixed together. Yeah, kind of. But that's what makes this a little bit more complicated. But you'll understand once you see a Punnett square. So remember, incomplete dominance is a blending of color. So let's look at and see how we write this on a Punnett square. So since we don't actually have a recessive allele, we're going to use two different letters today. Yeah, It's going to get crazy. So when we have a homozygous red, it's going to be big R, big R. When we have a homozygous white, instead of it being little r, it's going to be big W, big W. Because remember, neither allele is dominant over the other, okay? And then therefore, the heterozygotes that are the offspring of a homozygous red and a homozygous white would be a hybrid, which would be the phenotype pink, right? So RW would be the genotype and the phenotype would be pink, okay? So this is important to make sure that you understand when you're actually creating this Punnett square. And especially if it's a fill-in-the-blank test question, you got to use the right letters. Remember, we use the first letter of the dominant trait. Well, neither one of these traits or alleles are dominant. So we're going to use R and W, two separate capital letters, okay? So what type of offspring might be produced by two pink flowering plants? Now let's think back. Pink flowering plants, okay? What are the genotypes of the parents? Let's get that straight first, okay? So if you're pink, what's your only genotype you could possibly have? Well, big R, big W, and big R, big W. So let's set it up just like we would a monohybrid cross, okay? And this is a lot like Mendel's first experiment, right? And this would be crossing two of his F1 generation. So remember, he got all heterozygotes for his F1 generation when he crossed two true breeding plants. This is the same thing. But this time, since we know our heterozygotes have a different phenotype, pink, okay, our genotypes and our phenotypes are going to actually be the same. Okay, so if we look at our genotype, our genotypic ratio would be a 1 to 2 to 1. Okay, so 1 red, 2 pink, 1 white. And our one homozygous red, okay, 2 heterozygous pink, and 1 homozygous white. And for heterozygous, you really don't need to say heterozygous pink because we know that. But the phenotypes would also be the same, 1 to 2 to 1, 1 red to 2 pink to 1 white. And if we ask for probability, remember we want a percentage. So I would say, what is the probability of these two parents having a pink flower? And you would say 50%. What is the probability of you getting a homozygous red offspring? You would say 25%. Okay, so make sure you know the difference between your genotypes and phenotypes, but here your ratios are exactly the same, which is different than what we see in Mendelian genetics. So it's very interesting. Let's talk about codominance now. Remember, codominance is when we express both colors but individually. So when I look at a heterozygote, I should see both of those colors. For instance, a white cow and a red cow, all of their offspring will what we call roan. Notice what roan looks like. Roan is not only is it's like what a painted horse looks like. It's red, but it's also white. Okay? It's almost like polka dots. Okay, you can think about it like that. Remember, the way we remember this is through co-pilots, okay? 2 co-pilots. So in this case, codominance, both alleles are dominant, okay? And apparent in the phenotype of the heterozygous offspring. So both alleles are dominant, which is different than what we saw in incomplete dominance, which was neither allele was dominant, okay? So remember, here, codominance, both alleles are dominant, just like both co-pilots are actual pilots, right, and can fly the plane. So the real example that we like to use, and this is where it gets really interesting, guys. This is my favorite topic to talk about. I love blood type. It's so much fun. It's so interesting because it really relates to all of us. But we have 4 different blood types. Well, technically we have 8 if you want to include Rh factor, but we'll talk about that in a minute. But human blood types are codominant because we have this AB blood type. Now, what does all of that really mean? We're going to talk about it. So there's actually 3 alleles that actually determine blood type, and it's the A and B alleles, okay? And the O allele is recessive, okay? So we write them with I's. So it's going to be for the dominant alleles, the codominant alleles, that's big I superscript A, big I superscript B. And for our recessive, it's just little I. Okay, so now we got a lot going on, right? It's kind of like Mendelian genetics, but it's not like Mendelian genetics. It's like kind of complicated, right? Now, this I is important. This I stands for immunoglobulin. Now, what is all that? Well, you've heard of antibodies, right? Well, antibodies are part of our immune system that triggers our immune system to say, hey, this object is foreign. And it does so by connecting like a perfect little puzzle piece with what we call an antigen. Okay? Antigens are stationary and they're on our red blood cells. Okay? Antibodies are free floating in our plasma because remember, our blood is a heterogeneous mixture, meaning our red blood cells are solid and our plasma is our liquid, right? It's the plasma is what makes everything flow. Okay, so your antibodies are flowing in the plasma and your antigens are on your red blood cells. Okay, this is important to note because that's how we determine the type of blood that you have. Now, some blood types are codominant, some are recessive, like A, B, those are codominant. Okay, O is recessive. So how would we write that on a Punnett square and all that? Let's look at it. But before we really look at it at a Punnett square, I want you to think, okay, well, Miss Palizzi is type O. Well, Miss Palizzi's parents are type A and type B. So you got a couple of options here, right? We got 3 options. Either I'm adopted, my dad's not my dad, or my parents are what? Oh, they could be heterozygous, right? For type A. Because notice what you see here. You can be homozygous for type A, right? You can be heterozygous, meaning you're A and you got this recessive allele to give as well. For B, you have the same thing. B, you can be homozygous for B, or you can be B with this recessive O allele. Okay, and we write it with an I. If that's super confusing for you, you can put it on your Punnett square with B and O and A and O or A and A. That's up to you. But when you answer it on a test and when you answer it and when you see it on STAR and when you see it on AP Bio, if you move on to AP Bio, they will always have the I there. Okay. It also helps us distinguish that this is not Mendelian genetics. This is non-Mendelian genetics. So we know that codominance or incomplete dominance or something else is at play here. So that's also what it tells us. Okay. So the I is actually kind of important. But if it's really confusing for you to put it in a Punnett square like that, that's okay. But make sure when you answer it on a test or STAR or whatever, that you put the I there. Now, we can also have that B with that O recessive allele, right? So what do we know about Ms. Palizzi's parents? Because Ms. Palizzi is type O. Well, we know that both of my parents are heterozygotes, right? For type A blood, my mom, and type B blood, my dad. So we know that they have this genotype right here, these 2 right here. Now, what does that mean I have 2 younger brothers. Now, what does that mean for my 2 younger brothers? Could they be type O? Yeah, absolutely. It's 25%. But what else does that mean? It also means if you do the Punnett square between these 2 parents, A, heterozygous for A, and heterozygous for B, you could really come out with your children could be any blood type, right? So it's very, very interesting to see what blood types you get from a matchup like that or from a cross like that. Both of my brothers, one is A, one is AB. So, and it's a 25% chance for any of those, okay? Now, what does all of this mean? What does all that mean, right? We're going to skip this really quick, and then we're going to go back here. What it means is that if you're A, remember we talked about antigens, okay? If you're A, you have this little antigen A on your red blood cell. If you're B, you have this little antigen B on your red blood cell. Looks just like this, okay? And notice your genotype. You can be homozygous for A or you can be heterozygous, okay, for both of these. If you're AB, guess what? Since it's co-dominant, you have both A antigens, that's our little spike here, and B antigens, that's our little circle. If you're O, you have none. It's a completely clean little red blood cell. And for RH factor is just positive or negative. If you're positive, you're positive for a little RH factor protein or antigen on your red blood cell. Okay, so Ms. Pelosi is type O positive. Okay, so I'm completely blank with little RH antigens on my red blood cell. Okay, now we'll get back to what the RH factor positive and negative means here in a second, but I wanted you to see this picture so you understand what the different blood types actually mean and why AB is what we call it co-dominant. Okay, so let's go back to this problem right here. Already saw the answer, but let's go through it. Okay. What types of offspring might be expected if one parent has type AB blood and the other parent is heterozygous for type A? So what's the genotype of the first parent? So they have AB blood. So what's the only possibility they could be is IA, IB. Okay, and if you just want to write AB, cool. But remember, when you answer for a test or quiz, it must be like this with the I. What is the genotype of the second parent? Well, she's heterozygous, so hetero means different. So we know that she's going to have one recessive allele. So it's going to be IA and one recessive little I. Okay. So what are the possibilities? So let's set it up. I want you to go ahead and pause me and cross. Now, what'd you get? So what percentage of my offspring will have type B blood? So that's a phenotype. So I have 1. So 1 out of 4, that'd be 25%. Okay. What's my percentage of having an offspring with a homozygous A genotype? And that's 1, right? Now, so that would be 25%. Each one of these for genotypes, it's 1 to 1 to 1 to 1, right? They're all different for genotypes. Now for phenotypes, that's not how it goes. We have 2 type A blood, okay? Because even though this one's a different genotype, it still codes for type A, even though it's a heterozygote. And then we have 1 type AB and 1 type B. So notice here your genotypic ratio and your phenotypic ratio would be different. So it would be 2 to 1 to 1, okay, would be your phenotypic ratio, whereas your genotypic ratio would be 1 to 1 to 1 to 1. 1 to 1 to 1 to 1. Yep, that's 4. Okay, good. So we kind of understand how to work these Punnett squares. You have one also on your warm-up that will also be very helpful to you. So let's go back to this slide here. Now, I have a description of like who can donate to who, because that's important, right? Okay. Blood donation and knowing who you can donate to is super important. So in order for us to do that, we're going to skip through this a little bit, but we're going to go on to the next slide because I want to show you, we're going to start talking about antigens and antibodies. Okay. So for this one right here, this D, that is positive for RH factor. Remember we said that Ms. Pelosi is this one right here, O positive. That means I have this little RH antigen protein on my red blood cell, and that's all I have, okay? So remember we said antigens and antibodies, they're 2 different things. Antibodies are part of our immune system, and the antibodies connect perfectly with the antigen, and when they connect, the antibody tells the immune system that, hey, we discovered something. So here's where things get a little complicated, because we don't actually want our antibody to connect with an antigen, because then that would tell our immune system, hey, attack this red blood cell, right? We don't want to attack our blood, our own blood. That's an autoimmune system or autoimmune situation. We don't want that, okay? We don't want that at all, because that would pretty much trigger like clumping, what we call agglutination, and then you have clumping of your blood cells, and then that causes a stroke, and then you die. Okay, so that would be really bad. So what we want to do is not have the same antibody and antigen present. So let's talk about this, okay? And I'm going to skip through the RH factor for a minute, and then we'll talk about the RH factor and go back with that, okay? And remember, that's symbolized here with the D, the little orange thing here. So for A, remember, we're going to have your little A spike antigen, okay? And this one's A positive, so that also has the RH factor. It's going to have antibody B present in the plasma floating around. So can antibody B bind with antigen A? No, that's a good thing. We don't want that, okay? So the antibody present will always be the opposite. And it's part of the reason, we're going to talk about this, why only certain blood types can donate to each other, okay? So type A positive can receive from itself A positive, okay? A negative, okay? Because guess what? Negatives don't have this little RH factor antigen. So your body doesn't know that it's there or not there. It's always there, but if it's not there, it's no big deal, okay? So negatives can give to positives, but positives cannot give to negatives. So that's a good rule of thumb. Okay, I'm going to say that again. Negatives can give to positives, but positives cannot give to negatives. Okay? All right. So A positive, A negative, O positive, and O negative. Oh, whoa. Why? Because guess what? O doesn't have any antigens on that red blood cell. So there's nothing that this little antibody B can connect with. So we're good to go. Okay? And I want you to think about that right now. Okay. A positive can receive from A positive, A negative, O positive, and yes, O negative. Okay? So I want you to think about O for a second. Make a prediction. Okay, we're going to move on. I want you to see if your prediction was correct. So type A positive can donate to, okay, A positive and A B positive. Why? Because guess what? A B positive, right, doesn't have any antibodies present, so nothing's going to connect, and they still have that A antigen. It's good to go. Okay, so A positive can definitely donate to A B positive. Boom, done. Let's talk about A negative. So it's almost the same, but not quite. Okay, A negative has antibody B and antibody for RH, okay, because it doesn't have antigen B or antigen RH, okay, which is the anti-D. So we know that these two antibodies are present in the blood. So A positive cannot give to A negative, because these antibodies will then connect with these little antigens, and that will create an immune response. Your body will start attacking your blood. You will have a stroke, possibly, or a heart attack, or some kind of clotting, and die. Maybe not die, but I mean, depends on how much blood you received from that individual who was a positive. So it's a super dangerous situation. Blood typing is super important. Okay, so who can A negative receive from? Well, only A negatives and O negative. Okay, so remember, O is special because it's recessive. Okay, and it doesn't have any antigens on it. And O negative has 0 antigens on it. Okay, so that one, super special. We'll talk about why. So who can A negative donate to? Can donate to also A negative. So remember, negatives can donate to positives. Okay, so A positive, AB positive, okay, because here we are, right? AB positive, and AB negative because we're also a negative, okay? See how that works. Now, let's talk about B, okay? So if you're B positive, you have this little B antigen, and you have the RH antigen, which means your antibody that's in your plasma, which one is present? Anti-A. That's why A cannot give to B, okay? Because then this antibody will connect with this antigen. Clumping will ensue. What will happen? Heart attack, stroke, all that good stuff, okay? Not good. We don't want that, okay? So B positive can receive blood from, of course, B positive, B negative, because remember, negatives can give to positives, O positive, okay, because remember, O only has this little RH, O positive only has this little RH factor antigen, okay, and so does our B positive, and O negative, okay, because O negative, negatives can give to positives, but not the other way around, okay, so then who can B positive donate to? B positive and AB positive. So not very many, okay? And notice it's kind of similar to A positive as well, okay? So let's talk about B negative. B negative does not have the RH factor antigen, but it does have the B antigen. So that means that the antibodies present, there's two, antibody A and the RH antibody, okay? And so B positive cannot donate to B negative because that RH antibody will connect, clumping will happen, stroke. Okay, we don't want that. So the only people that B negative can receive from is B negative and O negative. Okay, you can donate to B positive, B negative, AB positive, because AB positive, look at this one. It's got this, okay, and it's got this, but hey, these match up, so we're good, and it doesn't have any antibodies present, so you're good to go, okay, and AB negative, because remember, negatives can donate to other negatives, all right? Now, let's talk about AB positive. AB positive is super, super special because it is what we call the universal acceptor. All of these blood types here can donate to AB positive and everything's good to go. So if you're AB positive, that's the best blood type to be, because if you're in a car accident and you're bleeding, if you're bleeding profusely and you need blood, we don't even have, if we know that you're AB positive, we don't even have to check any of the other blood. We just throw it in, okay? We don't have to type it. Because you don't have any antibodies present, there's not going to be any reaction to any of these other blood types when they go in. Your body's not going to see the difference, okay? Now, so you can receive from any blood type. Notice all the blood types are here, okay, all 8. Now, who can you donate to? Well, you can only donate to yourself or other people that are AB positive. There's the rub. But for you, it's super beneficial, okay? We're going to talk about the universal donor here in a second. So AB positive, super important to remember, star it, highlight it, whatever. It probably will be on the test. It is the universal acceptor. AB negative is almost the universal acceptor, but it can only accept or receive blood from all the negatives, right? Because positives cannot give to negatives, but negatives can give to positives. Okay, so we can only, since AB negative is a negative, we can only receive from other negatives, and the only antibody that it has in its plasma is the RH, what we call the anti-D here. Okay, so the RH factor. So who can it donate to? Well, AB positive and AB negative, of course. Okay, whoops, here we go. Now let's talk about O. O positive, that's Miss Palizzi's blood. And that's actually the most prevalent blood in our population here in the United States, and possibly the world. However, O positive is super helpful if you have O positive. They really like to have donations to that. So when you are, I believe you have to be, I think it's 18, is it 16 or 18 or older to actually donate blood. But I started donating blood when I was really young. And if you're a certain like height, weight. You can donate what they call double reds, and double reds is where they give you back your plasma, but they take all your red blood cells. Super helpful for people that need blood transfusions and what have you. And so why do blood banks want people with O positive blood to donate more? Because we're almost the universal donor. Almost. Okay, we can donate to any positives, okay, but we can only receive. Okay, sorry, I had to pause there for a second. So O positive can only receive from O positive and O negative, because remember, negatives can give to positive. But O is special because it can give to mostly, O positive can give to all the positives, A positive, B positive, AB positive, and O positive, okay? But it can only receive from O positive and O negative. Okay, so it's really cool that I'm O positive, and it is the most prevalent, I believe, in our population blood type. I don't know what is the most rare. I think is it AB negative? Is it AB negative? I think it is. You might want to do a quick Google search and double check me on that one. I usually memorize those. I didn't look it up again this year, but I believe it's AB negative is the most rare. So let's talk about our universal donor. And this one's kind of hard to see because my little thingy's in a way here. Can I move this? Okay, so my O negative, look at my red blood cell. I have no antigens on it, which means for antibodies, I have all of them. So I can't receive from any of these, okay? Because an antibody is going to connect with one of those antigens on any of these blood types. So I can only receive blood from O negative, but you gotta betcha that I can donate to every single blood type. So universal donor, it's awesome being the universal donor, especially if you like to donate blood, but it kind of stinks because if you're ever in a car accident and need a blood transfusion, I hope that another person who's also O negative has also been donated to the blood bank, because they have to be careful when they are giving you a blood transfusion, okay? Because you have all these different antibodies. And if anybody else from these blood types over here, any of these 7, your immune system will react with that, okay? So let's talk about multiple alleles. Many genes have more than 2 alleles. Genes with 3 or more alleles are said to have multiple alleles. We can also call that polyallelic. In genetics, problems with multiple alleles, there are 3 or more alleles for that trait, okay? So let's look at our blood types again. That's multiple alleles, right? Because not only it's A, B, those are 2 dominant, they're co-dominant, right? But we also have O. Okay, so we have at least 3 alleles that determine a genotype. We can also look at it like this, okay? So we can look at coat color and rabbits. So wild type is a fancy science word for what we normally see in nature. So when you see like wild type, you're like, oh, that's normal, okay? So, but scientists have to, think that they're fancy and call it wild type. But that's what you normally see in nature. So that's full color, often called agouti, okay, or wild type. C superscript CH, that's light gray or chinchilla, okay? And albino with black extremities or Himalayan, that's the phenotype. The allele is C with an H, or you have C albino, okay? So you have 4 different alleles here that create something completely different that we see, okay? So these 4 alleles just demonstrate a dominance hierarchy in which some alleles are dominant over others. The 4 alleles for coat color in rabbits in order of dominance are as follows. So agouti, that is, or our wild type, that's what we see in nature the most. That is why it's possible that that's because it's the most dominant. Okay. We also might have to think about environmental factors that play into that. Okay. Then second dominant one is light gray or chinchilla. Albino is the third one, or albino with black extremities, that's called Himalayan, and then albino is completely recessive. Okay, so polygenic inheritance, we're using that prefix poly again, so that means many. So you're gonna have many different genes, okay? Some traits such as size, height, shape, weight, color, metabolic rate, and behavior, they're not just determined by one pair of alleles on one gene. You're gonna have many different genes, maybe on different chromosomes. And that is why we experience such a spectrum of color, if you will, when we talk about eye color, hair color. Well, let's talk about hair color because hair color is messed up. Not only is it polygenic, it is incompletely dominant. Okay, so you can have a blending of two colors. You can have codominant, so you can have some hairs that are completely blonde or red. And in my case, I have like little streaks of red, and some hairs that are completely brown. Okay, so or blonde or what have you, right? So hair color is really very complicated. Eye color also very complicated. A lot goes into this. So trait polygenetic or polygenic, a trait affected by a number of genes. Okay, and it doesn't necessarily show a clear difference between each individual. So you're gonna, like I said, you're gonna see a spectrum of color. Skin color is like this. Okay, so that's why we see like a spectrum of light, dark skin colors, because all it really has to do with is the protein melanin and how much of it you have. Okay, so skin color is really interesting because you can have a parent with very dark skin and a parent with really light skin, and you might have a child with right in the middle, medium looking like darkness skin, right? And then you might have a child that is way lighter skinned, right? And it just really has to do with the different variations of all of those genes and how they come together and you express them phenotypically. It's very, very interesting, but it is quite complicated. Okay, let's talk about sex-linked genes. And before we do that, let's talk about our sex chromosomes. Okay, so you have two sex chromosomes. If you're a female, you have XX. If you are a male, you have XY. Okay, so we have some genes found on our X and Y chromosomes, and those are called sex-linked genes. Now, most of the time, our sex-linked genes really just help us with like producing the correct hormone for our gender type, that whole thing. Okay, and when we talk about gender, remember we're talking about science. So in science, it's not about like emotionally how you feel. It's like we're talking about genetics. Like if I ran your genes and I ran your DNA, what it would come out to be. Okay, so I'm just going to clear that up. So I'm talking about science right now. That's okay if you don't agree with me, but that's what we're learning in science today. So sex-linked genes, let's talk about it. So you have 23 pairs of chromosomes, and we talked about that. And let's look at our 23. Okay, we have 23 chromosomes pairs here. Okay, and notice we have an X and a Y here. So we have 22, what we call 22 pairs of autosomes and one pair of sex chromosomes. Our autosomes are what you would call your normal chromosomes that have all your genes that code for everything except for your gender or sex. Okay, so like I said earlier, males have an XY. That's what the males look like, and females have an XX. Well, it's actually very beneficial to be female. And I'm not saying that because I am a female, but it's because we have two X chromosomes, and there's a lot of genetic disorders that are found on the X chromosome and that are recessive. So females have to have two copies, or guess what? Males, you only have to have one. And we're going to talk about that today. All right, so one pair differs between males and females. This pair is called sex chromosomes. These sex chromosomes differ in the structure and contain genes that determine the sex of the individual. We usually see that in those chromosomes and how we produce certain hormones and proteins, and that really interacts with our phenotype and how we look. Okay, so we know that females look different than males. And females have two copies of the X chromosome, and males have one X chromosome and one Y. Most sex-linked traits in humans, like disorders, and most of them are disorders that we're going to talk about today, not just traits. Color blindness, hemophilia, muscular dystrophy, those are caused by recessive alleles, like I was talking about earlier. And all you need, if you're a male, is one copy of that recessive allele. So guess what? Color blindness, hemophilia, muscular dystrophy, and yes, men are more prone to male pattern baldness, right? Which is usually given to you by your mother. Not usually, it is, because it is on the X chromosome. And that is who determines who gives you their X chromosome. Because let's think about this for a second really quickly. Okay? Females inherit one X from mom, because mom can only give what allele? X. Okay? And one X from dad. Okay? So I want you to think about this for a second. Which parent determines gender? It is the male, because the male can either give an X or a Y. So if the male donates a Y to that offspring will be male. If that that father donates an X, that offspring will be female. So that's kind of ironic when you think about Henry VIII and we think about history and how he chopped off all his wives' heads because they wouldn't deliver him a male heir. Well, it's kind of his fault. Not really his fault. He didn't know what he was doing, and he had no idea that DNA even existed, nor did he even know that chromosomes existed or any of this existed. So is it really his fault? Who knows? Should he have done that? Probably not. Okay, so let's talk about color blindness in humans. So I want you to look at this chart really quick. And it looks a little complicated, but it's really not. So if we look at this, so we can see XX. We don't even need to look at this. We know, boom, female. 2 uppercase C's, which means this is a normal vision female, okay? This right here, this genotype still has a phenotype of a normal vision female, but she is a carrier for color blindness, which means that all of her sons will be colorblind, right? No, half of her sons, 50% of her sons will be colorblind, but 25% of her offspring will be colorblind, okay? And 50% of her daughters will be a carrier, okay? Now that really just depends on what the male is too, right? If you're a normal vision male, this is what your genotype looks like, okay? If you are a colorblind male, look at yours here. If you are colorblind, you only need one allele, okay? That one X, that's all you need, okay? That's why this carrier female, 50% of her sons will be colorblind, okay? 50% of her sons will be colorblind. Now, you could roll the dice each time you have an offspring. So could it be that if you're a carrier, you don't have any children that are colorblind? Sure, absolutely. But every time you have a new child or every time you have offspring, you roll that dice, okay? So let's talk about hemophilia. Hemophilia is a blood disease where your blood does not clot. So when you get a little scratch or a cut or whatever, you bleed profusely and you keep on bleeding. And so for that reason, way back in the day, okay, it was very hard for them to live past the 6 or 7 years of life. They didn't live for very long. And they were always usually very ill type children. They always kind of had major issues. So it's also a sex-linked trait, and it's recessive. So and it's on the X-linked. It's X-linked, so sex-linked, but specifically X-linked. And so males are going to be more predominantly affected here because they only need one copy of that little H, just like we saw in color blindness. And male pattern baldness is the same way too, and muscular dystrophy. Okay, but we are going to talk about hemophilia because actually the czars of Russia had a long line of hemophilia in their bloodline. And there's some reasons for that. Way back in the day, when you were royal, if you were of royal blood, you could only marry and have offspring with other royal blood people. So what that did was a lot of times you married a very close cousin, which that decreases genetic diversity. That is not good. We don't want that, because what happens, that increases your chances of getting genetic disorders like that. And what would happen is we would see a lot of females be carriers, and they would marry their close cousins, and then their children would have hemophilia, right? And we keep seeing this over and over, and we even see some of the females actually had hemophilia, but a lot of the males did too. And the czars of Russia, one of the last heirs to the throne that was killed during like the overthrow, and I mean, I'm not a historian, so but when Russia became the USSR, maybe I'm not on that same track there, but when they overthrew the Tsar regime, one of the last heirs, he had hemophilia. And so you see that we look at a pedigree and we can see how many generations and the different genotypes, and we can just determine who's the carrier and who's not. And we're going to look at pedigrees and karyotypes today. All right, so eye color and fruit flies, same thing. It's an X-linked trait. It's recessive. So if you have white eyes, that's a recessive trait. It's more prevalent in males. And so we know that it's an X-linked trait because we see that it's more prevalent in males. So, let's go back. A normal woman whose father had hemophilia married a normal man. So a normal woman. So first of all, she's a female. So we know her sex chromosomes are X and X, good. And so what's the H? So she's normal. So she's got at least one big H, but her dad had hemophilia. So he could only give her an X with a little H, right? So he was this, okay? So therefore she has to be heterozygous, and she is a carrier, and she married a normal man. So remember, men are XY, so he has to have a X big H Y. So this is how we set up the cross. That's the female, that's the male. So first of all, we see what percentage of offspring are female? 50%. What percentage of offspring are male? 50%. So if I ask what percentage of offspring are going to have hemophilia, you would say 25. Right? But if I asked you what percentage of male offspring would have hemophilia, you would say 50. So as you see here, I'm only going to have normal female individuals. Okay? Are you guys doing stuff? So like we talked about, color blindness is also recessive, and it's carried on the X chromosome. So it's going to be very similar to our other problems that we did. So you can have a bunch of different types of color blindness. I'm not going to go through all of this because we're at 53 minutes and 47 seconds, and that's a long time to be lecturing and you listening. So I'm going to move on quickly. So let's talk about pedigrees. I kind of mentioned pedigrees. It's kind of like a family tree, but we're tracking a specific trait that like is being expressed within the family through generation and generation. And if you're a circle, okay, then you are a female. If you are a square, you are a male, okay? If they're linked together like this, that they are married, okay? If they are linked together like up like this, they are siblings, okay? And these two, one blue, one brown eye, right? These two are the children or offspring of these two individuals, okay? And this individual is a female and married this male who's green eyes, and they had two children. One's female, one's male. When we are tracking a specific trait, we're going to shade in that circle or square, and that indicates that that person actually has that trait, okay? So if we're tracking hemophilia and this male is shaded in, that square is shaded in, that means he had hemophilia, okay? It's a really great way for us to be able to see through the generations how prevalent a certain, like especially if we're talking about a recessive X-linked disorder or even autosomal recessive disorder, disease, what have you. It's really cool to be able to see that throughout a family tree. Now, if you're heterozygous for a trait or a carrier, now you can color in half, because it really tells you like, hey, this person is heterozygous, but you have to know for sure. You can't make any assumptions when we're doing this. So let's talk about this practice problem right here. The following table shows 3 generations of guinea pigs. This is our first generation, this is our second generation, and this is our third generation. We usually label our generations with Roman numerals. So this would be I. Okay, so generation 1, generation 2, generation 3. We also label our individuals with Arabic numerals. That's a fancy way of saying regular numbers, okay? And we're going to start at the top left and we're going to work our way to the right. 1, 2, 3, 4. Then we're going to go down, back to the left, over to the right, 5, 6, 7, 8, 9, 10, 11. Go down to the left and then to the right, okay? 12, 13, 14. So it's important that you number these correctly. Notice shaded in individuals, okay, means that they do in fact have this smooth coat or this recessive trait, okay? So we can go through and we can actually label all of those because we know those genotypes for sure, right? Okay, because based on their phenotype, we know they're all small r are homozygous recessive because we know that this is a recessive trait. Now, do we know what number 2 is? We can make some assumptions, but we don't assume in pedigree check, okay? So, but what we do know, none of these individuals, these two, their children, their offspring, excuse me, none of them have a smooth coat. So, but dad did. Therefore, dad could only give a little r. So what are all of they? They are all heterozygous. We know they're all heterozygous here, okay? Because dad could only give a little r. And they don't have a smooth coat. Now, this person, we can say, okay, hey, they're heterozygous for sure. We're not sure what number 2 was, and we will never be sure. If they had an offspring that did have a smooth coat, we would know for sure that this one was heterozygous, but we don't have that. We don't have enough information. So we have to put both of these genotypes here. Now, number 8 individual, married number 9. They are both heterozygous, and they have 2 children that have smooth coats. Now look at number 3. Do we know? We don't know, okay? Probability would say that probably would be heterozygous, but you really cannot be for sure. Number 11 is the same way. We really cannot be for sure. We are sure for number 9 because 14 and 12 are both recessive, okay? So we can really look at our offspring here, determine our offspring's phenotypes, genotypes, and then look at our parents' offspring, the parent genotypes and phenotypes. So, and know for sure their genotypes. Very interesting. So, autosomal recessive traits, usually you see them skip generations. Males and females will have it, okay? And most will have carriers. So most will be carriers when you see stuff like this. So I1 and I2, what can you say about them? Well, they have to be carriers, right? Because they have an offspring that has that autosomal recessive trait. What can you say about 2-4 and 2-5? Okay, they also have to be heterozygous because look, they had an offspring with which is 3-4. Okay, what are the odds that 3-5 is a carrier? Well, the odds that 3-5 is a carrier would be, these are two heterozygotes, so it'd be 1 to 2 to 1, so it's a 50% chance that it's a carrier, right? Because remember, it's still a 50% chance that, it's only a 25% chance that 3-4 would actually get that autosomal recessive disease, and it did. So you remember, it's 25, 50, 25 every single offspring. So for 3-5, that would still be 25%. All right, so X-linked recessive. X-linked, your big thing here, it's also going to be rare in the pedigree. It's also going to skip in generations, but you're going to see more males affected than females. Now, I'm not saying that you won't see a female, but it'll be like these 3 males and maybe like 1 female, right? Okay? You'll see more males affected than females. Okay, so you can kind of see these trends. Remember, if it's a square, it's a male. If it's a circle, it's a female. Okay, if they're joined like this in the middle, they're married. Okay, if they're joined from up top like this, they're siblings. If it's shaded in, they have that trait. So let's look at this one for color blindness. So look, individual 2 is colorblind. It's a male. What do we know about males? Well, they only have 1 X, okay? So it has to look like that. And both of his sons have that, okay? Now, let's look at all of our individuals here. Okay? This one, number 5, we know she, because it's a female, because 2 X's, right? And she's a circle. She does not have, she's not colorblind. However, she is a carrier. Okay, and that's because dad could only give her an X with that recessive allele. So she has to be a carrier. And because dad doesn't actually pass down these X's, the XB, that X allele to his sons, okay, mom actually passed these down to them. Remember, dad having this, that's just a coincidence. Dad does not usually, we do not usually see a dad pass them down to their sons because dad does not pass down the X. Mom only pass downs the X. If dad passes an X and mom passes an X, it's a female. If mom will always pass an X allele. If dad passes a Y, it is now male. So remember, the male does the determination here. Now let's look at 7 and 8, and they have these 4 offspring here, 11, 12, 13, 14. So 7 is a male with color blindness, that is colorblind. 8 is a female that is a carrier, okay? So they're going to have, let's see, well, if mom passes an X to her sons, that's little, okay, they will be colorblind. She did not. So 50% of her sons will be colorblind, but in this one, they're not. None of their sons are colorblind, but she does have a daughter that is colorblind because dad passed a recessive X and mom also happened to pass a recessive X, and that's a 25% chance. And we also see the last daughter here. She's also a carrier, okay? So let's talk about karyotypes because these are pedigrees, family tree looking things. Karyotypes, it's a picture, snapshot of a person's chromosomes, and we match them by pairs and arrange them in order from big to little, okay? And we match up the bands and their length, okay? So that's how we get your homologous chromosomes all matched up. And the DNA is taken during mitosis. So extracted, you take a picture of all the chromosomes, and then you kind of piece them together. It's quite interesting. So let's look at it. Reasons for karyotyping, genetic disorders. So when you talk to a pregnant woman, a lot of times they're like, hey, I need to go get genetic testing done. This is the kind of stuff that they're doing. They're checking for all these genetic disorders. And you're like, but why though? A lot of these genetic disorders can really limit lifespan. So that you really need to prepare the mother for that. It can dramatically change the family dynamic. okay? Which is not a bad thing, but your family needs to be prepared for that. So, and giving them this opportunity to, and you don't have to do DNA testing or chromosomal testing or anything like that for genetic disorders, but a lot of couples and females that are pregnant opt to, because it is good to prepare for whatever is coming in the future. And so if you have a normal, healthy child, that's amazing. That's great. You don't have to, like, you just have to like actually be a parent, which is like really hard work. No, thank you. But those parents with children with these genetic disorders, it's even more difficult. So what are the pairs? They're homologous pairs, and we're going to match them up by length here. Notice big to small, and then your last pairs right here, those are your X and Y. Those are your sex chromosomes. So we have autosomes, okay? Those are your normal, control all of the traits that make up you besides your gender, and that is chromosome number 23. That is your sex chromosomes, okay? XX and females, XY and males. So if you look at this one right here, we have all 22 autosomes, and here are our sex chromosomes. So this is a normal, yes, female, good, because we have 2 X chromosomes. This one is a normal, look at this one right here, Y. And notice it's normal because we have 23 pairs, okay? When we start seeing more than 23 pairs, like on chromosome 21, call that a trisomy. If you have more than 3, that codes for what we call a disorder called Down syndrome. And I show you a picture of a child like this because they usually have a webbed neck. They have like different facial features, larger forehead. They also have slower like mental development. And a lot of times they don't live past the age of like, I believe it's like 35. It might be longer now because, you know, technology. But it used to be around 35 back then, back when I was like learning about this. So I should probably double check that. But it is caused by a trisomy on chromosome 21. And you're like, but Ms. Polizzi, like, how did we get an extra chromosome in there? Like, what happened? Let's talk about meiosis. Okay, remember in meiosis, what happened? Our chromosomes all aligned, and then it split, right? And then guess what? That happened again. Chromosomes all aligned down the middle, and then they got pulled apart, right? By our little microfibers, right? Our little spindle fibers or whatever they're called, right? Spindle fibers. Miss Pelosi should use our big science words. Okay. So sometimes they don't attach appropriately and correctly. And when they pull them apart, they don't get pulled apart. So one egg or sperm or gamete gets an extra chromosome, okay, and one gets one less. So now we're talking about 50% of your eggs or sperm that you just made or gametes that you just made are now kind of messed up. So if you get an extra and it's on chromosome 21, that codes for Down syndrome, and it's called a trisomy. And that not being pulled apart correctly during meiosis is called non-disjunction. And so whenever we see an extra chromosome, so this one has not 46, okay? No, not 46, 47 chromosomes, okay? Yeah, brain explode emoji. And that's a lot of DNA. That's a lot of extra DNA in there, okay? Because that's a whole chromosome. And then you have the other gamete, right? Or one less chromosome. That's a lot of DNA that you're missing. Like Klinefelter's disease is XO. They're missing an entire sex chromosome. That has a lot of effects on how their development, okay? Their physical development. And we're going to do a whole genetic disorders research project so you can research your own genetic disorder, and you'll pick one from at random, and it's going to be really interesting. I can't wait to see what you guys come up with. So this is just a little bit of what I told you about, Down syndrome, non-disjunction, all that good stuff. So what I want you to do now, because it's like an hour and 10 minutes, and this is a long lecture. Sorry, guys. I get really into it. But what I want you to do, your homework for today, besides your 2 worksheets that you need to be doing from class today, your pedigree one and your blood typing one, I want you to go home. I want you to ask your parents, what's your blood type. And then I want you to ask your parents what's their blood type. And I want you to draw out a little Punnett square and see what are all of the possibilities of offspring your parents could have and what percentage are you. Okay? Because we know Miss Pelizzi's, my mom is A, my dad is B, I'm the 25%. Actually, my brothers are also the 25% because my parents could have any of the four major blood types. Okay? So, super interesting topic today. I hope you enjoyed. Bye.